In this video, we're going to be examining the Watson and Crick DNA model. Watson and Crick developed the structure for DNA in 1953, and we still use it today. We'll start by looking at the four nitrogenous bases in DNA. These bases are thymine, cytosine, adenine, and guanine, and they're split into two groups, pyrimidines and purines. Now, pyrimidines have one cycle group, while purines have two. Now, when we go to pair these bases up, we see that thymine binds with adenine and cytosine bonds with guanine. And why is that? Well, it has to do with the hydrogen bonds. Thymine and adenine each have two hydrogen bonds, while cytosine and guanine have three. Now, for this reason, we won't see an adenine bonded to a cytosine or a thymine bonded to a guanine. Now, we also won't see two pyrimidines bonded together or two purines bonded together for the same reason. Additionally, if we had two purines bonded together or two pyrimidines, this would cause an uneven DNA structure that would not function normally. Now, these hydrogen bonds are weak, but they ensure that the right bases pair up together. And now we'll look at the other molecules involved in the structure of DNA. Here I've drawn out the nucleotide structure. In the structure we have a phosphate group, a deoxyribose sugar, and a nitrogenous base. The deoxyribose sugar has 5 carbons, 1, 2, 3, 4, and 5 prime. On the first carbon we see that this is where the nitrogenous base binds. On the second carbon we'll either see a hydrogen or an OH group. A ribose sugar would have an OH group, while a deoxyribose sugar will be missing an oxygen. On the third carbon we'll see that this is where the next phosphate group would attach to, right on this OH group. And then finally on the fifth carbon, this is where the previous phosphate group attaches. Let's say that this strand is part of a double helix. This is what the other half of the DNA would look like. If you look closely, you will see that these strands are running anti-parallel. This means that the phosphate group bonded on the 5' carbon on this end is facing one way, while the 5' carbon on the other end is on the other side. Same thing with the OH group. These strands are anti-parallel so that they can be replicated in the correct order. Okay, so now we have a completely bonded DNA molecule. As you recall, the phosphate backbone had an overall negative charge. This charge causes the strands to repel from each other, while the hydrogen bonds and the bases act to stay together. This causes the twisted helix that we see in DNA. Now, if we place the DNA in a highly concentrated salt solution, we'll see that the positive sodium ions will neutralize the phosphate backbone. This allows for the DNA to become stabilized and as a result becomes more tightly wound. Now we'll see that the bases are still stacked in the same order and it is still right-handedness, right? Because these are going up and to the right. However, the major and the minor grooves you're seeing will become more tightly wound. As a result, more energy would be required to unwind these strands for DNA replication. And because more energy is required, this would also increase the TM, or the point at which DNA becomes single-stranded.